The goal of this procedure is to quantify the response of flies to the social signal emitted by stressed individuals. This is accomplished by first collecting responder and emitter flies no more than 2 days before the experiment. The second step is to transfer both emitter and responder flies to fresh vials 2 hours before the experiment and let them habituate to the environmentally controlled room where the assay will be performed. Next, the responder flies are placed in the tea maze and given the choice between a vial in which flies have been previously stressed, thus containing the Drosophila stress odorant, and a fresh vial. The final step is to count the number of responder flies in the fresh vial, in the DSO vial, and in the elevator to quantify their response to the DSO. Ultimately, the tea maze is used to measure the avoidance by flies of a social stress signal, specifically the Drosophila stress odorant.
I first had the idea for this method when I was a postdoc at Caltech. At that time, Greg Sue and his postdoctoral advisor, David Anderson, were interested in studying fear or social avoidance in Drosophila, but there was no existing assay for that. They approached me because I was performing a lot of behavior assays in Seymour Banzer's lab. And while performing phototaxi and olfactory learning experiments, I had noticed that flies were attracted to vials in which other flies had spent some undisturbed time, while avoiding vials in which flies had been stressed, at that time with an electric shock.
Visual demonstration of this method is critical as the experimental process of the social avoidance assay requires some practice to be quick and efficient in transferring the vials without losing any flies or DSO.